Good morning and thank you for coming. It's our privilege to serve cancer patients today in 2014, particularly because we can see things more clearly than we've ever done before. If I could sing, I would sing, but I won't. I think genomics today is really elucidating the etiology of this disease or these diseases that impact our families and our loved ones and maybe even ourselves. But we are able today to give hope to more patients through trials that are being presented at this meeting. I am so excited that we have trials at this meeting that are showing significant benefit for diseases where I never dreamed we'd see this kind of benefit. Melanoma. I never thought I would see drugs work in this disease. A decade ago, hardly any drug was available that had any impact on this disease that is becoming increasingly common and frequently sunshine associated. So this is a time of hope, but there's also much work left to do. A lot of the work that is emerging is really fueled and driven by essentially our ability to see what previously was white light becoming a multitude of beautiful colors. We can see more clearly the precise genomic aberrations that actually are hijacked by these cancer cells and drive tumor growth. And that is allowing us to move from being very, non-selective, non-specific in how we target these cells to the holy grail of cancer medicine that is specifically killing tumor cells and sparing normal cells by hitting the Achilles heel, these genomic drivers that drive tumor growth. Now that may sound very feasible, and it is, and that's why we're seeing a major improvement in diagnostics and therapeutics, but there is a major challenge in that these cancers are genomically complex, they are highly adaptable, they diversify with the selective pressure of the treatment that we give these patients. And we have to increasingly consider not only inter-patient heterogeneity, so my disease that I focus on, prostate cancer, is not one disease but 30, 40, 50 diseases, but also intra-patient heterogeneity and different clones that are causing resistance to the drugs we use in one patient, and considering optimal drug combinations. And you'll hear at this meeting, for example, for BRAF mutant colorectal cancer, a disease where very few drugs really have a major impact, emerging evidence both scientifically as well as clinically through a rational trial design, how we are having a major impact on this common subtype of this common disease, colorectal cancer. And what you'll see at this meeting is that increasingly our trials are being designed differently, and therefore allowing us more leverage on really understanding the disease, but also delivering patient benefit. And the challenge today is to give treatment more precisely, to give better care, to cause, less side effects, and to try and help more patients more effectively with better quality of life and longer longevity. So our shared vision for our society is better patient outcome, more efficient healthcare, faster and more cost-effective drug development. And increasingly this is going to require utilization of smart genomic biomarkers, not only from tissue biopsies, but increasingly from blood-based sampling of tumor DNA. And there is a whole session on this, which my colleague and friend Charlie Swanton is chairing, and I think this is so exciting. My group have published a paper on this quite recently in Science Translational Medicine. We can now access through liquid biopsies information on cancer genomics through a blood test. So these are key figures. You can see these here. We have a large number of abstracts. We have a substantial number of late-breaking abstracts, some of which I am confident will very soon change practice. Many of which will change practice in the not-too-distant future. We have 265 program hours of sessions and 459 speakers. The trials are divided into 100 phase 3 trials and many phase 2 and 1 trials involving more than 125,000 patients. This is truly a magnificent and global effort. And at least a third of our abstracts, probably more, involve a focus on precision medicine and utilizing patient selection through these predictive biomarkers, through genomic analysis of tumor DNA, or protein, or RNA, to help us deliver treatment better. Some of the highlights, and this is not comprehensive. We haven't mentioned bladder cancer, prostate cancer. There are major presentations for those diseases here too. I think, to be honest, this is probably, in some ways, a melanoma meeting. We have some really spectacular results for melanoma, and this is so exciting for me. We have some amazing sessions on immunotherapy. Immunotherapy has arrived and will be part of our armamentarium in cancer medicine going forward. We have spectacular data on colorectal cancer emerging, although still early, but I am very confident it's going to really impact the field in the not too distant future. We have exciting data for lung cancer, for different subtypes of lung cancer. So this is a time of great hope. Our patients are waiting, but there's still much more left to do.